Hi there, Dr. Inch here again. Welcome back to my kitchen. Okay, today's experiment, what we're going to be looking at is extracting the DNA from fruit. Okay, so as you will remember, the DNA is in the nucleus of the cell. We are going to break open these cells and extract the DNA from there. Okay, so what we're going to use an apple. You can use other fruit as well. Kiwis work really well, strawberries work really well. If you haven't got something to mash this up properly, it might be easier using a kiwi because they're a little bit easier to mash up. Okay. The reason I'm using apples is we've got apples here. They've been in the house ages, they haven't been eaten yet. So rather than waste them, I was going to use them for an experiment. Okay. So, first thing we need to do, we need to get rid of the seeds, we need to get rid of the stalk, and we need to get rid of the skin. So in the best possible condition, here's one I made earlier. So what we need to do, we need to get that cut up into chunks, because now we're going to break open as many of these cells as possible. What you will need for this experiment, you'll need your apple, you'll need a bit of water, you'll need some salt, you'll need some washing up liquid and a bit of a cloth or a dish towel or a sieve of some sort which has got a reasonably fine mesh to it. Okay. You might need some kitchen roll as well, but I found that you don't necessarily need to do this. So this is something that I have done hundreds and hundreds of times. Not like this. But in the lab, I used to extract DNA from bacteria on almost a daily basis. This time, what we're going to do, as I said, is do it from an apple. So first thing we need to do, we need to add a couple of teaspoons or tablespoons of water. And then we need to add a tablespoon of salt. All of this is going to help break open our cells. Let me add just a little bit more water there as well. Okay, so we need to get all of this mashed up. Might take a little bit of time. Probably should make it a little bit smaller to start off with. Make sure you get it all mashed up. This is the first step of breaking open the cells. Let me put that down there so I can mash it up nicely. There's still some big bits in there, but what we've managed to do, we're managing to mash that up nicely. Oh, look at that lovely apple paste. Okay, so that is our first stage. That's the mechanical breaking down of the apple, and we're starting to damage the cell wall. Remember, it is a plant cell, so it has a cell wall. Our second step is now to add washing up liquid. Doing it in the lab, you use slightly more specialized chemicals, but effectively it is washing up liquid. So what we're going to do, we're going to add about a tablespoon worth of washing up liquid. It's nice and runny. What we don't want to do, we don't want to get lots and lots of bubbles, but what we want to do is get that all mixed in nicely. So give that a gentle stir. And what the detergent will do, or the washing up liquid will do, it will start to burst these cells and will release everything that is in the cell into the liquid. So what we do now, we leave that, we let that sit for about 10 minutes. Sometimes you can heat it up a little bit, use warm water rather than cold water, but that's up to you. You can mess around with that and see how that works. So what I'm going to do is I'm going to pause that now for 10 minutes and let those cells all start bursting and releasing all of the cytoplasm and all of the other organelles into the liquid. So I'm going to pause right there. Okay, so that's our 10 minutes up now. And we'll see how good my editing skills when I come to link those two videos together. Right, so we've now got a bit of a gloopy liquid. If you look at that, I have agitated it a little bit just to make sure that it has been working, but I've been very careful not to produce lots and lots of bubbles. So now what we need to do is we need to filter it. I'm just using a kitchen cloth. You can use a dish towel. Using the kitchen roll like we did last time isn't going to really work because it is quite thick and it's quite gloopy, and that will just sort of block up. So what we need to do, we just need to pour it onto here. As you can see, it's a little bit runny. Let that soak through. Might need a little bit of help. If you're having problems going through, all you need to do is add a little bit of water. I'll show you why it's struggling to get through in a second once I've filtered it. We just need to encourage it along a little bit. But again, something that's quite difficult to do when we have got using kitchen roll. Okay, so let me add a little bit more. Now it's starting to get quite gloopy. But I can see it running through. I'll show you it in a second. I'll pour it into a different glass in a second. Right, while that's draining, what I'm going to do, I'm going to get my hand sanitizer out of the freezer. It is really important that you have your hand sanitizer as cold as possible. Like I said, in the freezer for at least an hour. Mine has been in there for overnight, so it's been in there 24 hours. Okay, so let me just get a little bit more through of this. That's coming through nicely now. So obviously the more you collect, the more DNA you can collect as well. Okay, let me get a different glass so we can actually see what's happening, because this jug is not particularly brilliant. Let's get this last bit in here. There is lots there. That was quite a big apple. Okay. As you can see, it's quite lumpy. It's quite gloopy. This is why we need to try and filter it out. Right, so let's get rid of that. And let's put it into a different glass so you can actually see what's happening. Okay, so there we go. There is still quite a lot of washing up liquid in there. That's what's giving it its nice green colour. But that's not going to make a huge amount of difference. So let me get rid of that and let me grab my hand sanitizer. Okay, so there's my hand sanitizer, just a standard hand sanitizer. It does have to have alcohol in it or it's not going to work. We then need a smaller glass and we pour a little bit of the solution into there. Now what we need to do is we need to get the DNA out of there. So what we're going to do is we're going to add the hand sanitizer and we're actually going to run it down the side of the glass and get a decent level of liquid in there. Let's just make sure the side of the glass is clean as well so we don't get any mixture. Okay, so like I said, what we're going to do, we're going to run the hand sanitizer down the side two separate layers forming. Okay, right. I'm not sure if you can see that. So we have our two separate layers there. Okay. It'll take a few minutes to work, but I can already see there are some cloudy strands coming up into the hand gel, if you can see those. Okay, so what we'll do, we'll just put that aside. What we might be able to do, let's see if this actually works. See if we can get it. See if we can see that. Okay. You see those stringy bits that are starting to form? That is apple DNA. The DNA is insoluble in alcohol, which is part of the hand gel. So we're getting a precipitate of DNA. The longer you leave it, the more you will get. Okay, have a go at that. It's brilliant fun. As I said, I used to do it on a almost daily basis in the lab, and this is only the second time I've ever done it in the kitchen. The first time was just to make sure that it worked. Have fun with that. It's a really, really cool experiment. So thank you for joining me in my kitchen, and I will see you soon.